Okay, so yesterday when I did the video for you to take notes, it was so long, so I want to redo it and try to compress this, and that way you can stop and start it wherever you need to take notes. So the cytoskeleton. Since I've already written the notes out, I'm just going to read and kind of elaborate as we go. So for the cytoskeleton, think protein. Cytoskeleton is what's going to obviously give the cell everything from shape to help with intracellular transport, to help even cells divide, all types of things. But usually, in the first year class, we just don't talk about it a lot. And that's probably the fault of the teacher because it's something that's not emphasized on our exam. So it's time to kind of change that. So here we go. So the cytoskeleton is made of different types of proteins. They can be filament-based or they could be in the form of a tube. And we're going to notice there are 3 different sizes of these types of filaments. They do produce a network through cytoplasm and beneath the cell membrane. And so you're going to see them located in different places and they'll have different jobs. Generally speaking, we want to make sure that if you have a general question about the function of the cytoskeleton, here are general functions. Please be careful about listing because they want some elaboration. So you can elaborate by telling the type or stating which type does these things. If it's a question that specifically is asking you to compare the 3, then you need to go in greater detail. Pay attention to what they're asking. If it says identify, you just basically list it out. If it says describe, you have to give more support. If it's compare, you tell differences and similarities. Okay, so here we go. So generally speaking, the roles of the cytoskeleton provide shape and support to cells. I've drawn out a little red blood cell, a neuron or a nerve cell, and a little paramecium just to show that the cells differ in shape. And what helps to keep that shape in place can be the cytoskeleton. Okay, they organize and anchor organelles. They aid in the cell division, everything from the formation of the spindle fibers in that complex to actually helping to produce a cleavage furrow in cell division of animal cells. Also in cell signaling, which we'll talk more about later when we talked about cell-to-cell communication, and they assist in the movement of these vesicles we keep talking about within the cell. Okay, so all cells, no matter if they're prokaryotic or eukaryotic, have a cytoskeleton. However, as in cases, in most cases with the other structures, the prokaryotes do have a less complex type of cytoskeleton. Okay, so without further ado, let's go back up to our diagram here. So I have a visual of the 3 types of 3 components of the cytoskeleton. The first one, AF, I learned is microfilaments. They're actin filaments. They are the smallest of the 3. They're made up of little actin monomers, and typically in circumference or in diameter, actually, it's about 7 nanometers, which is 10 to the negative 9th. Intermediate filaments, you can see what they look like in terms of size and structure. They are made of about 50 plus different proteins. One example would be keratin, and they're about 10 nanometers in diameter. So when you look at those, we'll write down some more details about their shape in just a sec. Okay, and then the microtubules are hollow. They are made of alpha and beta tubulin, and they can be as large as 25 nanometers in diameter. Okay, here's another visual of those so you can see how they vary, but they're not in the same order here. So I like putting the intermediate filaments in the center because they are kind of intermediate, even though they're made of different things. Okay, all right, so actin filaments, still talking about structure. They are more flexible, so they allow for that. Intermediate filaments, They're there to reinforce and give a cell some strength and some kind of, think of scaffolding kind of. And even the microfilaments can do some of that too beneath the cell membrane. And microtubules are more rigid. Okay, so let's look at these here. So microfilaments, you can see the size. As far as the composition goes, we said these are globular actin proteins. They're polymers of those twisted in a double helix. Functions of the microfilaments are in cytoplasmic streaming. So you remember the little video that I posted from last week, and it showed an amoeba and it was surrounding the paramecia, and that movement is helped along or just assisted by this these microfilaments. So it's going to help the cell membrane to morph and move. And also you can see here in this diagram that I put with the amoeba here and a white blood cell there, and they are actually bringing those large particles into their little cell by something called phagocytosis. So this movement, cytoplasmic streaming, it also is done in plant cells. Also between, remember those little holes, those plasmodermata, and it allows for movement of that cytoplasm between adjacent plant cells. Okay, we have membrane support, so it's highly concentrated beneath the cell membrane, and you'll see a diagram in just a minute of that. They also have a contractile component to them, which is noteworthy because they are going to work with a motor molecule called myosin. So actin and myosin work together with muscle contraction. And so we're going to look at that a little bit further on Monday. Okay, and then there are also, these are really thin tracts that run throughout the cytoplasm too, so they help with some movement of vesicles. And the word microvilli actually is associated with these cells here. So these are stomach cells, and the microvilli are these little folds. Okay, so microvilli, I've written it on that one. And so this increases surface area, of course, when you see anything that's folded, and it's going to allow for more digestion, absorption of those monomers that are digested. We also have some villi in the small intestine. So here are the microfilaments, or these actin filaments, that are really supporting, and they're hugged up and really close to the cell membrane. Okay, and they can be attached to the cell membrane. Alrighty, so that's microfilaments. So intermediate filaments, they are rope-like, and it looks like several of those kind of twisted together. Here's something that's very important here is there's no motor component to these, so they are more fixed and permanent. They anchor those organelles and keep them in place, and again, that offers some support within the cell also. Okay, so a diagram of that is here. And here, so it's adding some, they said mechanical strength. Right. And lastly, microtubules. So microtubules, they are made actually of a series of alternating alpha and beta monomers, and they are aligned, and several of these are aligned together, and then they form this hollow tube of tubulin polymers. So let me show you that here. So alpha and beta. So alpha and then beta. And you can see how they alternate. So each one of these is a little strand. Well, for some reason it's not going to work. Uh-oh. Sorry. All right, let's see if this is going to work better now. Sometimes it cooperates and sometimes it doesn't. Okay, so here's your strand, and this is one of these, okay? And you can see how it's hollow. Now, what's interesting is, think of these as like railroad tracks, and there's two different directions. We have these motor proteins that are going to carry vesicles, kind of like if you are carrying something over your head and you're walking along a track. And so you'll see that kind of in a diagram that I'm going to show you. But the direction that they travel depends on which end of the microtubule they're moving to. Okay, so the alpha end is the negative end of the microtubule, so they're polar. And the beta end is the positive end. Okay, now that's going to be relevant because we look at the direction that these things travel. Okay, so let's talk about the movement of vesicles first. So this intracellular movement, we have the two different ends. We have two motor proteins are going to travel along those tubes. So if it is the kinesins, that is the motor protein, they're going to always move the vesicles towards the periphery of the cell or towards the cell membrane. Okay, so think about those will be moving from the Golgi out to the cell membrane, or from the Golgi out into the cytoplasm, farther out to the to dump a lysosome out there. It could mean that they're moving a vesicle from the rough ER to the Golgi, or from the smooth ER to the Golgi. So what you have to know here is that they always move towards the positive end of the tubulin. So if this is cell membrane here, this would be a beta tubulin end of the tubule. Here's the alpha tubulin end, which is closer to the inside. So we'll just put a nucleus over here. Okay, so these things would be traveling this way, carrying the vesicle. All right, and those are kinesins. The dynin motor proteins are going to keep vesicles moving towards the center, so towards the negative end. So we have those are going to be moving this way. So that is its role in movement. Next, we have its role in cell division. And so with cell division, we know that microtubules actually make up the centrioles. They also make up the spindle fibers that manipulate the chromosomes during cell division. So they move them to the center of the cell during metaphase, and they separate them and move them away from the center during anaphase and then telophase. So they play that role. And then lastly, they make up cilia and flagella. And so I want to show you this arrangement of what these look like. So again, here's the alpha and beta. And you'll notice that they're in these little dimers, and the dimers just mean there's two of them. And there's the alpha and beta. So they're arranged kind of like, they're opposite each other. So it's like kind of like the positive and negative are together, which makes sense. And this is one dimer, and if you count all the way around, there should be 9 of those. and then there's 2 singletons in the center. So we call this a 9-to-2 arrangement. So this is something that cilia and flagella have in common, that they have that. And then the next video is going